The building foundation of genetically modified foods is that all you're doing is adding one trait, one gene to create one protein to create one trait, and everything else is fine. In reality, when you do that blasting, you can create all sorts of problems.
In some areas of the world, the introduction of genetically modified foods has been met with a lot of controversy. Some countries, such as Venezuela and Hungary, even ban the growing and import of certain genetically modified crops out of fear that they will cause adverse health effects such as cancer. But are such concerns legitimate? For generations, farmers have made use of hybridization to grow bigger, better yielding, and more profitable crops. Today, scientists make use of genetic engineering techniques to accomplish the same goal. Unlike hybridization techniques, which requires the crossbreeding of the same or closely related species, genes from organisms ranging from bacteria to animals can potentially be extracted and introduced into crops. Genetic engineers were once interested in splicing genes from a species of cold water fish with tomatoes to make frost-resistant crops. Although this fish tomato never came to be, it raises a lot of questions. Would this affect the natural ecosystem? Would it be safe for individuals with fish allergies to eat? Would the insertion of such foreign genetic material alter other properties in the fruit, possibly to the detriment of the consumer? The reality of the situation is that it's a very complex one. The trait of interest is hardly ever the only trait affected.
There was a gene chip that monitored gene expression in the DNA at a time when the insertion occurred. Compared to prior to insertion, 5% of the host's genes were disrupted, meaning that they either increased, decreased, shut off entirely, or shut on entirely their production of protein. So if something is pumping out proteins at random that wasn't supposed to, what could that mean? This list will come up several times tonight. It could be allergenic, toxic, carcinogenic, anti-nutritional.
In the mid-1990s, world-renowned protein scientist Arpad Pusztai studied the effects of feeding genetically modified potatoes to rats. These potatoes were engineered, like many crops today, to produce their own insecticide. The natural insecticide that was transformed into the potatoes was the protein lectin. These lectin-producing genes were taken from the snowdrop plant, an organism that naturally produces the protein. The rats in this experiment were separated into 3 groups, 1 experimental group and 2 controls. The experimental group was fed potatoes that were genetically modified to produce their own lectin. 1 control group was fed normal, unmodified potatoes, while the other was fed potatoes that were injected with lectin extracted from the snowdrop plant. This second control group was necessary to distinguish between observations that were contributed to the lectin itself from those due to the genetic modification process. Rats in the experimental group developed a number of negative traits by the end of the feeding study. These included stomach lesions and gastrointestinal inflammation. Interestingly, this was not seen in either of the control groups. This led Arpad and his colleague to conclude that the negative traits were not due to the lectin itself, but rather due to something that occurred during the genetic modification process.
The problems were that the genetically modified potatoes induced what we call a proliferative growth in the small intestine, and I shall explain what it means. But before I do that, the most important thing was that we pre-selected the gene that its product should not do that. So we spent 6.5 years of selecting out a gene whose product wouldn't do the thing which we did see in the genetically modified potatoes.
Needless to say, these conclusions sparked some debate. Critics of Arpad's study believe that his study was flawed. The rats in this study were only fed potatoes and no other form of nutrition. They claim that using an experimental group of rats that were solely fed genetically modified potatoes is not a practical, real-world example. Regardless, the only obvious difference between the two groups exposed to lectin is the genetic modification process. As Arpad concluded in his study, the observed negative effects on the rats resulted from the way the gene had been inserted into the potato genome and what it did to the potato genome. The study was not conducted long enough to see cancer-causing effects, but taking into account the observations of proliferative growth, changes in cell metabolism, gastrointestinal ulcers and inflammation, the potential is there. Unfortunately, there was no follow-up study. In fact, Arpad Pusztai's controversial conclusions led to him losing his job and reputation, as well as having all his data seized.
I thought I had complete control over my new ice cream flavor, Stephen Colbert's Americone Dream, so imagine my surprise when I recently read this off the carton. We oppose recombinant bovine growth hormone.
The issue of injecting cows with recombinant bovine growth hormone, or RBGH, is occasionally a hot topic in the media. This hormone is produced by a genetically modified form of E. coli. When injected into dairy cows, it has been shown to significantly increase milk output, but at the expense of the animal's health and fertility.
Now, rather than just tell you about all the cool science I learned, I'll let the video speak for itself. Jimmy?
A Van Dyke, Yao Ming, and menopause. What do all of these things have in common? They're all produced by hormones. Meet Farmer Joe. His cows aren't producing much milk. Luckily, he can help nature along with a genetically engineered growth hormone called RBGH, which evidently you shoot up a cow's butt.
The issue as to whether or not growth hormone in milk affects humans is up for debate. Studies have shown that RBGH is indistinguishable by the human gastrointestinal tract from natural bovine growth hormone, leading the FDA to deem it safe for human consumption, regardless of whether the levels are higher than in the natural state. Furthermore, the FDA found that 90% of RBGH is destroyed during the pasteurization process anyway. However, many argue that this data was deliberately falsified.
It's a great time to be a high-producing cow. Posilac One Step, bovine somatotropin by Monsanto.
The federal government basically rubber-stamped it before they put it on the marketplace. The longest test they did for human toxicity was 90 days on 30 rats.
Posilac is the single most tested new product in history and is now available to you specifically so you can increase your profit potential.
And then either Monsanto misreported the results to the FDA or the FDA didn't bother to look in depth at Monsanto's own study.
And this former journalist is not alone with this belief. There are many scientists who argue this as well.
We have been pressured and coerced to pass drugs of questionable safety, including the RBST. I personally was very concerned that there's a very serious problem of secrecy, conspiracy, and things of that nature, and something needs to be done.
Canada, along with many other countries around the world, have outright banned the use of RBGH on dairy cows.
Fortunately, America's not one of them, thanks to one of our country's bedrock principles: innocent until proven cancerous.
But assuming this data is correct, that RBGH, as it is present in the milk, does not pose a significant threat, it still offers no evidence that other factors in the milk of cows treated with RBGH are safe for human consumption. And not surprisingly, milk from such cows differs by other factors as well. More recent studies have found that, along with an increase in growth hormone, there is also an increase in insulin-like growth factor 1, or IGF-1, in the milk. An FDA study found that this increase is very small and likely nothing to be concerned about. But IGF-1 is recognized as playing a vital role in the development of many tumors due to its anti-apoptotic activity. Many current studies have been looking at the possible link of IGF-1 to a number of cancers.
Two very sophisticated journals published this information in 1998. Premenopausal women with high levels of IGF-1 are 7 times more likely to develop breast cancer. Outside of family history, it's the #1 risk factor. Men are 4 times more likely to develop prostate cancer. IGF-1 is also implicated in colon and lung cancer.
This is significant because IGF-1 found in milk is identical to that found in human beings, which leads us to ask, are people who drink milk more at risk for developing certain cancers?
Milk and hormones. As natural as milk and cookies. And hormones. Brought to you by the Prescott Group. We mean business.
The evidence suggesting genetically modified foods are harmful to human and animal health is extremely limited. Although there are far more studies saying that these foods pose no risk, their conclusions are sometimes very narrow-minded. Many of the studies only seem to test the safety of a single compound found in the food, ignoring the fact that the majority of genes are pleiotropic in nature. Typically, the altering of one molecular process has more than the single desired effect. Considering the growing cancer rates worldwide, one would think there would be a plethora of studies looking at the health effects of something as fundamental as food consumption. Although it is not widely perceived as a threat in this country, there are millions of people in other parts of the world who actively avoid eating genetically modified foods. The lack of solid evidence on either side of this important debate accomplishes nothing but the fueling of conspiratorial suspicions involving corporate greed and hidden agendas. This only makes the legitimate claims regarding the potential dangers of genetically modified food appear less reputable. There is a definite need for more testing to be done. Along with animal feeding studies, human epidemiological studies should be conducted to specifically look for potential links between ailments and the consumption of genetically modified foods. Until then, much of the debate remains hearsay.
Remember that all these studies are done before you can do any experiments with humans. Now, in this case, of course, the whole thing has been changed around because you're being used as the guinea pig. They are doing the experiments on you, and what is even more, you don't know whether they are doing it or not, because in the US, for example, it's not labeled, so therefore you don't know whether you're eating GM food or not.